a step back from multicellularity, but not all the way back to prebiotic chemistry, so right in the middle, are all of us microbiologists trying to understand the diversity in these tiny unicellular organisms. And originally, I guess I got involved in microbiology or interested out of curiosity. But in the context of astrobiology, understanding the whole diversity of how single cells can make a living is important to understand or choose proper targets for exploration beyond ocean worlds. Also interpret the findings from the data that we get back from our explorations, and perhaps also in the future, once we, if we understand how the breadth of these organismal niches, we can prevent planet cross-contamination by preventing organisms from one body going into another. So in order to do this, we need to understand what is alive and how microorganisms make a living. And essentially, this is something that Olga already explained, but if you're looking for something that is alive, that grows, reproduces, and undergoes evolution, that organism needs to be able to do one fundamental thing, which is obtain energy. And the difference with us, that we depend on oxygen, microbes, prokaryotes, can employ a wide variety of substrates to get energy. And what that shows, the redox tower shows, is that as long as microorganisms can figure out the biochemical strategy that allows them to take something, to transfer electrons and harness the energy from the transfer of electrons from something that has a low redox potential to a higher one and transform that into ATP, then they can use the energy to perform a wide variety of functions and survive. And on Earth, we have a huge number of niches, tiny and big, and all sorts of microorganisms employing different strategies. And our task as microbiologists, in a way, is to understand each one of them and see the potential for each one of them. Even though if on Earth they are in very small niches, they could be predominant in other planets. But we not only have this huge planet to explore life, we have 4 other Earths to explore respiratory diversity. Perhaps not in the geographical space dimension, but in time. As Marcus already showed, the Earth, our Earth, went through different transition periods over time that depending on oxygen levels, driven in turn by microorganisms. And in each one of these scenarios, the Earth had completely different environments that probably select for different types of organisms. At the beginning, where there was no oxygen, the atmospheric composition was also very different, probably full of methane or hydrogen. And so different microorganisms that perhaps are not very abundant now probably were very abundant at the beginning in the Proterozoic or Archean worlds. And also the oxygen levels drove different other electron acceptor levels, such as iron and manganese and other substrates that microorganisms could incorporate for life and to develop different strategies to breathe and generate energy. And I am interested in particular in iron and manganese. And we know the broad trend of iron coming down from millimolar, micromolar levels to nanomolars, in which nowadays iron becomes even limiting for life. And manganese is also probably following a similar trend, and both can be used by bacteria to harvest energy. And nowadays, there are very small pockets of the Earth where these ancient conditions prevail. And we call them, some of them have very similar characteristics, such as high iron, low oxygen. And we call these ancient ocean analogs, like the one picture that you see there. So our strategy, if you want to look at potential primitive mechanisms for life, not only now but also in the past, is to explore these ancient ocean analogs and search for the metabolisms that we're interested in. So that's what we did. We trying to understand the different forms of manganese cycling in the environment. We took samples from Lake Matano, which is the ferruginous high iron, very low carbon ancient ocean analog that has a manganese cycle. As you can see there, it's likely driven by microbes that oxidize and reduce manganese, which ends up precipitating. But geochemists have looked at these at this lake for a long time, and so it's very well characterized in terms of redox. We know that it's permanently anoxic, and iron and manganese are big electron donors, acceptors, but we don't know much about who's doing what and who is responsible for the manganese cycling. So the strategy that we employed was to take sediments from this lake, cultivated under pre-GOE conditions. A lot of iron, no oxygen, with the selection pressure of adding only manganese as the electron acceptor. And over time, we hoped that we would isolate organisms involved in manganese cycle, and we would have pure, beautiful cultures, cells. And we would transfer them over time to get rid of all the sediments and other components to obtain microbes that only use manganese. So what we saw after several transfers over around about a year is that, I wish I had, oh, there it is. This essentially shows the community structure, who is there, and what we use is the 16S, the ribosome, rRNA genes as a phylogenetic marker to extract the composition of the whole community and see who is there. And the most important point of this figure is to show you that rhodocyclaceae and Bolcoliales, these two green and blue bars that you can see after a few transfers, but not on the original inoculum, appeared, and they are both from the same group of bacteria called betaproteobacteria. So we wanted to try to understand why those were enriched in our conditions. So we wanted to dig deeper into what were they doing there and who they actually were. So we took the whole set of complete sample of DNA and proteins together, and we did metagenomics and metaproteomics. What this means is that on one hand, we took the DNA is sequenced into very small fragments are sequenced, and then bioinformatically they're put together into bigger chunks, eventually maybe making a complete genome, depending on your sample. On the other hand, we took the complete set of proteins, also the little peptides were passed through LC-MS, and that pattern that is generated was compared back to a theoretical pattern from the translated nucleotides from the DNA. And that way you can see which proteins are expressed. And we did that with the help of Brooke Nunn, Dr. Nunn from the University of Washington. So what we saw that these Dechloromonas that we saw were enriched, also we were able to get the whole genome from the community. And we compared it with known Dechloromonas species. We confirmed that this belongs to that genus. And we compared the genomes, the identity of the genomes to the one most closely related to it, the most, like the closest relative. And we found that they were 85% identical. So to assign two different organisms into the same species, you need at least 95%. So with 85%, we concluded that this was a new genomic species. That was enriched in our culture, but we weren't able to isolate it yet. So we're calling it Candidatus Dechloromonas occultata because it's still hidden in our samples. It was hidden originally in Lake Matano, and but what we did, what metaproteomic revealed us was that it was responsible for 73% of all the peptides that we found, which means that even though we didn't really thought that this was the most predominant or dominant member of the community based on that one genetic marker, it was the most active. And the rest of the DNA was probably from dead cells or bias from the PCR, or the method itself that bias is biased depending on how many copies of that 16S RNA gene you have. So then I'm not going to go into detail, so don't worry, but we were curious to see what attributes, what lifestyle this bacteria has. And we were curious about two things. One, their ability to transform metals. And another thing that called our attention was the presence of a denitrification pathway that was active, even though we hadn't added any substrate to express this pathway, but that's another story. But back to the multi-heme C-type cytochromes. This is probably a case of structure determines function that we just heard. So the one thing that we rarely pay attention to, besides the redox potential of different substrates that organisms use to make energy, one has to consider the availability, how the organism sees it, and how available it is to them. And even though manganese and iron are pretty good electron acceptors, closer to oxygen than sulfate and sometimes nitrate, they depending on the redox state of the metal, they're going to be found in solid or soluble form. So when organisms reduce it or oxidize it, they're going to either find it in an oxide form, so it means that they cannot get it inside the cell like we do with oxygen or nitrate, some other organisms, or they're going to produce an oxide, which means that is going to precipitate, and organisms probably do not benefit from precipitating things inside their cells. So in both cases, whether an organism is oxidizing or reducing, somehow very interestingly, they use the same type of machinery, in a way, biochemical machinery. They use this electron conduit that is made of cytochromes. In the case of metal reducers, it's called MTR, metal reduction, and in the case of metal oxidizers, it's called MTO or PIO, depending on whether the organism is phototrophic or not. So we saw, for the first time in this type of species, that this organism had this gene, this conduit, that all these genes were together in one operon. And we couldn't find it in any other Dechloromonas. As you can see from this graph, these are two copies. If you put them in the context of a tree with closer organisms that also have these conduits, which is important because if we had just tried to look at the gene on its own in the complexity of the community, we wouldn't have known it belonged to the Dechloromonas. The reason we know it's a Dechloromonas gene is because we mapped it back to the metagenomic bean that we obtained. And this gene and the architecture of this operon is also present in two other beta-proteobacteria that are uncultured. So this whole branch here are uncultured organisms that have this same operon. Also, this conduit is not present in the only other Dechloromonas that is a known iron metal oxidizer under anoxic conditions. So this is pretty unique and new to this novel species. Another somehow correlated operon that we saw is this cytochrome C-rich operon. As you probably saw from other talks, the cytochromes are really good, heme groups are really good at transferring electrons. So whenever we see multi-heme cytochromes in an organism, we typically can very confidently say that they're doing some sort of electron transfer outside of the cell, especially when there are decahemes, 10 hemes or more than 10. So again, we saw this undecaheme that was present only in this species, this new species, but not the other Dechloromonas relatives, again present in uncultured beta-proteobacteria, rhodocyclades and gallionellales, which means that they must be doing something that we haven't figured out yet. Some of these that are cultured and that have this operon have also been shown to be involved in metal cycling. And we were also surprised to see, this is just three examples of this whole genome. Nitrous oxide reductases, which means that is part of the denitrification pathway. It's not really important to the story, but what is important is that this genome, this strain, as opposed to the other ones, was highly enriched in sensors, including in this nitrous oxide reductase operon, which was pretty unique compared to the others. Which means it may use, one thing that I did not mention is that the reversibility of this conduit has been seen in many organisms. For example, metal reducers, when you can reduce also electrodes, if instead of a metal you have an electrode, but depending on the redox potential of the electron, you can make the electrons go back. So, and many organisms change their metabolism depending on the redox potential of their environment. So the high concentration of redox sensors in metal reduction pathways coupled to nitrogen could be an indication of highly versatile, flexible metabolism of these beta-proteobacteria in these unique environments. So I'll just wrap it up by saying a couple of conclusions from this short story is that doing this enrichment and putting this selection pressure allowed us to magnify the existence of this novel conduit, which we wouldn't have seen by targeting the whole metaproteome from the whole community in situ even, because the concentration of this population was very small, the abundance. At the same time, because we enriched, we were able to capture a genome by genomic binning, and that constrains who we assign this function to. If we had just looked at a bunch of genes from the community in situ, we would have probably mapped it to a completely different species. But now here we have it in the context of a whole organism, and that's what we're looking for. We're trying to understand the evolution of organisms. And this probably is a product of lateral gene transfer, but we were able to map it straight onto this one species. And also, thanks to metaproteomics and metagenomics, we can see that this whole operon, not just one particular gene, but different operons, we could see them again expressed on different samples from different locations. So now, even though we were not able to isolate one species, we can collect all this information, and perhaps that will guide us to further efforts in isolation or understanding what can they be doing as opposed to having one single cell in the lab. And I'm still working on trying to isolate these beta-proteobacteria because we do need to characterize biochemically everything that we see from our metaproteome and metagenome. And I also would like to thank the whole astrobiology community. It's very inspiring to be here and listening to all the talks. And Jen Glass's lab, but also other mentors that I've been having for the past 2 years, like Chris, Reinhard, and Yang Zi, and all our funding sources too.